So today I'm going to talk about the work that we've been doing on applying these deep learning methods to analyze biological systems with single cell resolution. And so before I start, I did want to say that all the work that I'm presenting today here has been a really awesome collaboration with Michelangelo and Liat Karen up at Stanford. I know Liat's going to be on the job market pretty soon, so if anybody's listening, keep an eye out for her because she is amazing. So before I get started, I always think it's a good idea to step back and sort of, reflect on like where fields are at and what the challenges have been and what's keeping us from getting where we need to be. And so, today you're going to hear a talk about applying like all these different what used to be considered like difficult techniques to various problems in biology. But why bother to do that in the first place, right? Why can't we just do simple stuff and get like the knowledge we want out of these different biological systems? And that really is this question that's on the screen: why is biology hard? And in my view, biology is hard for three reasons. So one, there's a large parts list, right? If you look at a simple organism like E. coli, it has on the order of 3,000 different genes. You can consider one gene like one part. And when you consider mammalian organisms like people, then look at like the number of genes like a surrogate, then you're starting to get at the order of tens of thousands. And then there are a variety of different ways that mammalian organisms are able to get even more parts out of that parts list. And so any notion of understanding of how these living systems work, they have to be able to speak to this complexity that they have large numbers of parts. The second reason is that living systems are both dynamic and also spatially heterogeneous, right? So they change in time and they also change in space. And again, any notion of understanding a living system has to capture this aspect of the complexity. We have to understand how things change in time, and we also need to understand how things are changing in space. And so the third reason why living systems are hard is that basically the way they're composed of these objects called cells, which are this fundamental unit of living matter, and every cell is a little bit different, right? And so if you think of, say, a lung, you have the airway cells, you have the epithelial cells, you might have immune cells there, and each cell has a different function. Each one is composed of different molecules, and these differences actually matter for the functioning of the overall unit. And what that means is that because these differences matter, our best statistical techniques for dealing with large and noisy data sets, i.e. averaging, they all kind of get thrown out because the averaging are going to remove the very differences that you care about. And so this is a challenge, right? How can you make sense of these complicated systems using tools that give you large noisy data sets when in a very real way you can't average? Fortunately, for each one of these challenges, we have a technological revolution that's occurred over the last 10 years. And these revolutions are just fundamentally changing how we study living systems and the types of information that we can get out of them. So for the issue of having a large parts list, we have genomic technologies. And so these are a set of tools that really let you get at what is the composition of living matter. And so you can look at which genes are present using DNA sequencing. You can look at which genes are being turned on or turned off using RNA sequencing. You can look at what proteins are present using different proteomics technologies. And these tools are now getting so powerful and so sophisticated that you can even do these sorts of analyses in individual single cells. So I mentioned briefly about the issues of having temporal dynamics and also spatial heterogeneity. And for those, we have revolutions in imaging. And I think for a lot of people in life sciences, everybody would agree that our ability to probe these systems using imaging technologies has never been greater. We can look at single molecules, we can look at single cells, we can look at entire tissues. And these imaging technologies give us a handle on, looking at both the dynamics, but also how things vary in space. And lastly, to deal with the fact that we can't average, we have these new set of tools from machine learning. And so these tools really let us get insight from large noisy data sets in a way that respects the nuances of biology. And so the really exciting thing about working in the space now is that all of these different technological revolutions, they're starting to talk to each other. And so, for instance, you have different technologies that have genomics talking to imaging. And so these technologies will let you look at gene expression in a way that preserves the spatial information that's present in your sample. You'll have merging between, or communication between imaging technologies and machine learning technologies, and that's primarily what we're going to talk about today. So I did want to talk about one of the examples that of a merger between imaging and genomics. And so this is a merger between imaging and proteomics. And so this is a technological platform that was pioneered by Michelangelo and Liat Trent up at Stanford. And what it lets you do is it lets you look at up to 50 different protein markers within a clinical specimen simultaneously. And I just wanted to walk through how it works, because these are the sorts of data sets that we've been applying the machine learning techniques to. And so the starting point is you have a tissue of interest that you want to look at. And so for the work that's presented here, these tissues come from 41 patients that have triple negative breast cancer. And so then you have a set of proteins that you want to look at. And so for each protein, you get a molecule called a antibody. And so these antibodies can specifically recognize each of the individual proteins. And so for each antibody, you then label it with a metal ion. Yeah, and so each different antibody here colored in red, blue, and orange, each one gets a different ion, and these ions are basically like heavy metals. And so the key thing is that each antibody now has a different a mass tag. And then you take your panel of antibodies. There's a lot of work that has to go into making sure that these antibodies are actually measuring the things that you care about. But then once your validation experiments are done, you basically take your antibodies, you stain your sample, and then you go off to get it quote unquote imaged. And so because the antibodies are labeled with a heavy metal, your imaging is now imaging mass spectrometry. And so what happens is you take an ion beam, and so your ion beam then scans over your sample. And so at each spot that it sits on, it vaporizes that area. It releases a cloud of smoke. Within that cloud of smoke are all of the different antibodies that were labeled in that region. And that cloud of smoke then gets sucked up and sent off to mass spectrometry. And the mass spectrometry can then identify which of the different mass tags are present at this area, in this area that you're scanning. And then that gives you that basically gives you a signal. So for each antibody that you're looking at, you can basically ask, okay, in this location, how much of that antibody is present. And so that's how it works in one location. And so then to get an image, you basically take your ion beam and then scan over your sample. And then for each spot, you ask, okay, well, how much of each antibody species is in this spot, is in this spot, is in this spot, is in this spot. And then as you scan, you basically build up an image of what this well, you basically build up an image of where each of your different markers is. And so this is a great technology. It works with up to 50 different markers. It actually works with a fixation method called formalin fixed, paraffin embedded. And this is how many pathology departments preserve tissue specimen. And there are a lot of things to love about this technology, but one of the issues is that the data analysis is actually very hard. And so we'll talk about it later. And so I did want to walk through one of the case studies that this technology has been being used for. And so that's trying to quantify interactions between the immune system and tumor cells in triple negative breast cancer. And so for those of you who've been paying attention to sort of the oncology space, you'll know that there's a revolution happening where people are sort of figuring out how can we get the immune system to recognize cancer cells as foreign and use this as a modality of treatment. And there are different ways that people are doing this, but they basically come in two flavors. One is to basically take a part of your immune system out of your body, reprogram it using genetic engineering so that it will recognize cancer cells as foreign, and reintroducing it. Another way is to basically remove the brakes that already exist on the immune system. And that's more or less like what I'm going to talk about here. And so removing the brakes, this is called sort of inhibiting immune checkpoints. And there are a number of checkpoints that people investigated, like CTLA4, PD-1, and PD-1 ligand. And these checkpoint inhibitors have shown significant promise in numerous clinical trials. Yeah, so this is great because this is basically giving hope to a segment of patients who didn't really have hope before to even qualify for these sort of treatments. You already have to have failed multiple rounds of chemotherapy. And so while they have these therapies have a lot of promise, there are issues. And so one of the issues is that there's just a large variance in who responds. And so when you actually go through the trial data and look at what fraction of people respond, it's on the order of regular chemotherapy. You can expect roughly 15 to 30% of patients to actually respond to a given immunotherapy. And this is problematic because these therapies aren't benign. The risk profile for them is actually, for having severe events, is actually very similar to regular chemotherapy. And approximately 10 to 20% of patients will have toxicities that are so severe that they have to be taken off of the therapy. And so this is a problem, and ideally what one would like to do is precision medicine. Can we identify which patients are going to have a good response, which patients are going to have a poor response, and can we identify which patients are likely to tolerate the therapy and which patients are likely to have a severe adverse event? So to do that, to have this predictive understanding, really what you'd want to know are basically like, what are the immune tumor interactions that are present for each patient. Unfortunately, there's just a lot that we don't know about how the immune system interacts with these tumors, and we can put, one can put their physicist/quantitative biologist hat on and just ask some simple questions of what are some things that we'd like to know that we don't know. For each patient, and these are listed here. So some of the things that we might want to know: who is there? What are the immune cell types that are present within any given patient's tumor? Moreover, how many of them are there? Do you have a lot of T cells? Do you have a lot of B cells? Do you have a lot of macrophages and whatnot? You'd also like to know who are these immune cells talking to? Are they sitting next to other immune cells to get additional instructions? Are they sitting next to cancer cells to be reprogrammed into being inert? What do their interactions look like? We'd also want to know what does the expression of these immune checkpoint markers look like? What cell types do they live on, and are immune checkpoint markers co-expressed? So there are actually a number of trials going on where people are trying to test multiple immune checkpoint markers simultaneously, and looking at and having some information about co-expression would presumably help guide those studies. And lastly, what you'd like to know, what are just the general tissue features that predict who's going to survive, and what tissue features are going to predict who has a response? And so what you see from the end of the talk, for some of these questions, we have answers, others we don't, but at the very least, we sort of have all the different technologies in play now where we can get these sorts of information. All right. Okay. And so to get this information, what one has to do is get a cohort of patients and put together an antibody panel that will tell you the sorts of things that you want to know. And so that's what Liat and Michael did. They put together a panel of antibodies that would give you some information on what the tumor is doing. And so these are antibodies against different tumor markers. There are antibodies that will tell you what the different immune cell types are. And so you'll have antibodies that identify lymphocytes. You'll have antibodies that identify monocytes, neutrophils, and whatnot. You'll also have antibodies that will give you information about the state of the cell. And so, for instance, we see here there's an antibody against double-stranded DNA, and this will essentially mark all the nuclei of different cells, and we can actually use this nuclear information as a signal for cell segmentation. But there are other markers here too. So Ki67 is a marker that identifies replicating cells. these H3K9 and H3K27. These are markers that will tell you some of the status of the chromatin. There are also markers for the different immune checkpoints that different people are interested in, and then also markers to identify stromal cells. And so then, what they did, so they got a panel of tissue biopsies from patients. They put together this antibody panel, they validated it, and then they used their multiplex ion beam imaging platform. Basically, that's what the spatial proteomics experiment is called. And then basically looked at the expression of all these different markers in the tissues. And so I don't want to spend too much time on this, but this is basically some of the data, what some of the data looks like. And as you can see, it's exactly as advertised. So here are 4 different patients. This dsDNA channel is identifying all the nuclei, and each of these different markers are identifying a different biological aspect of this tissue. And so really what this means is that we, for almost the first time, you can get high resolution 50 channel images of these clinical specimen, where each channel gives you information about something that's biologically meaningful. The question then is, how do you analyze it? And the quantification really comes down to doing single cell analysis. Ideally, what we want to do is go into these images and then identify all of the individual cells that are present. And then once we know which pixels correspond to which cell, then look at those different markers and then aggregate that information. And so for each cell, we could say like, okay, how much P53 is it expressing, how much of the immune cell type markers are expressing, and then use that information to classify cells into different types and different states. And so we might be able to say like, hey, this really big cell over here is a cancer cell. The cell over here might be a T cell, and it's expressing like these immune checkpoint markers. To even get started with that sort of analysis, you have to identify where the individual cells are. And this is actually a major challenge with single cell image segmentation. The conventional computer vision approaches aren't really robust enough to segment images like this, just because there's significant heterogeneity in what different nuclei look like. And this quantification is essential. Pathologists have inter-operator differences. On 3-color images, it's kind of unreasonable to expect them to be able to interpret 50 color images. And digital pathology is really the only reliable solution. Fortunately, this is where deep learning can come to the rescue. And so there are a great set of techniques. So there are a great set of techniques for doing image segmentation and object recognition in images now that come from the deep learning space. And I just want to walk through like one of the schema that we use for doing image segmentation. And this is really thinking of image segmentation as image classification. And so if you think about a image classifier and how it works, it's basically a black box, and you spit in images and it'll spit out a label. So here we have images of a cat, a dog, and a chair. We spit into the black box, and out comes a label. And it says like, okay, I think this is an image of a cat, this is an image of a dog, and this is an image of a chair. If we had an image classifier that was able to do the following, then I'd argue that the image segmentation problem is roughly solved. And so let's say we had an image classifier that could go into an image, and for every pixel, open up a small area around that pixel, and use that local information to make the following determination. Is that center pixel either inside of a cell, at the cell, part of the background, or at the boundary between either the cell, the inside of a cell and the cell and the background, or at the boundary between 2 cells? If we had an image classifier that could do that, then I'd argue the image segmentation problem is solved, because then you could then go run your classifier over your entire image, classify every single pixel, and then use the following output as a segmentation mask, if it worked 100% of the time. The only question would be, what is that classifier? And that classifier is, these are deep convolutional neural networks. And so their mathematical structure is sort of inspired by human vision. And so when you think about how human vision works, here's an example of facial recognition. Our visual system works by first identifying low-level features. And so here's identifying edges. And then these features get grouped, right? And so you recognize edges, then collections of edges, then collections of edges, until eventually you're identifying high-level features. And so here, these are images of, well, basically features that look like eyes, features that look like a nose, features that look like a mouth. And then at the final level, your brain says like, hey, I see something that has 2 eyes, a nose, and a mouth. I think I'm looking at a face. This is very much what many deep learning, deep convolutional neural network architectures look like. You have sets of mathematical operations that extract features, like convolutions. You'll have mathematical operations that will basically introduce nonlinearity, so you can learn nonlinear relationships. And then you'll have downsampling operations that move you from a fine spatial scale to a coarser one. And so this is basically going from edges to collections of edges. And the key thing is that these functions, they're all learned. And so these weights of these different convolutions are variables that get learned during a training procedure. And so what the solution now looks like is very much software 2.0, where software is where the data is in a very real way, in a real part of your software. You start with a training data set where an annotator has labeled what's the inside of a cell, what's the cell boundary, and what's the background. You then go and train a deep convolutional neural network to do your cell segmentation. You then can apply that neural network to new data sets and then derive insight from those segmentation masks. And here, essentially what we do is use those segmentation masks to query all the different channels in our image. And then for each cell, we can look at what the different markers that are being expressed are. And then we can start using clustering algorithms to group cells in different cell types and cell states. I did want to show, so I do have to say, like, this approach is significantly more accurate than all of the other conventional classical computer vision techniques and also other machine learning methods. And this improved accuracy really lets us do quantification, both of cell type, but also of cell numbers in these pathology specimen. I did want to show where we're heading with this, and so we've actually gotten things to the point where with the figure 8 engine we can actually do things on human in the loop. And so what we do now is we start with our original image. We have an expert annotator in-house who annotates a collection of images, and this gets used to train a deep convolutional neural network. That neural network will work great most of the time, but sometimes it'll make mistakes. And so what we then do is we use this neural network to sort of highlight new data sets, and then we merge the original data with the prediction from our neural network, and then we feed that into the crowd. And so the crowd then goes and takes these images, and so here we have the raw data with the annotator or the neural network and highlighting. And the crowd then goes and takes you from the 90% of the way there to 100% of the way there. And these new annotations can then be used to train new neural networks, and the cycle can continue. And so what these approaches let us do is we can start with images like this, and we can then add a layer of semantic information, where here we've just highlighted all the different immune cell types that are present. So we've highlighted the B cells in yellow, regulatory T cells, neutrophils, macrophages, et cetera, et cetera. We're getting to the point now where we've accumulated enough of these sorts of data sets where we can actually train neural networks to do both the segmentation and the cell type classification simultaneously. And so this is an image sort of like showing a pilot data set where we've done that. And yeah, and so I know I'm short on time, but I just want to briefly walk through some of the analyses that we've done. So what you can start to do is just quantifying the immune cell type populations that are present across different patients. And so for these 40 patients that we looked at, we're basically able to quantify how many of the immune cells that are there, what fraction are T cells, what fraction are natural killer cells, B cells, macrophages, et cetera. And moreover, how does this immune cell type representation differ between patients? And not surprisingly, there's a lot of heterogeneity. Exemplified between these two patients. Patient number 16 has every immune cell type under the sun. Patient 24 just has macrophages. Moreover, it looks like there's actually a hierarchy in immune cell recruitment. And so this is basically a patient level analysis. And what we see, and so on the y-axis are patients, on the x-axis are the different immune cell types. Yellow indicates that this immune cell type is present, and blue means is absent. And so basically what you see is a hierarchy. And so patients that have natural killer cells will always have B cells, T cells, and macrophages. Patients that have B cells, they may not have natural killer cells, but they'll always have T cells and macrophages. Similar story for T cells, and macrophages are present in every patient because they're present in every tissue. Moreover, we can also do similar sorts of analyses with immune checkpoint markers. And so here are basically analyses showing what cell types immune checkpoint markers like to live on. Here, so LAG3, for instance, likes to live in this cohort of patients we looked at, likes to live on regulatory T cells and CD8 T cells, whereas EDO likes to live in natural killer cells and monocytes. And moreover, we can actually look at co-expression of immune checkpoint markers in single cells. And so we can ask, okay, for a single cell, which ones like to express PD-1, which ones like to express EDO, which ones like to express both. And we can do this for every pair of immune checkpoints markers that we looked at. And so this is doing a patient-level analysis, looking at which immune checkpoints are just present or absent for patients. And so what we see is that there are some patients that will have every immune checkpoint marker. There are some that will have none, and there's some that have a mix. But the really important thing is that now with this technology, we're actually able to identify who belongs into which group, which patients have all the immune checkpoint markers, and which patients have some, and which patients have none. So we can also do spatial analyses, and so we can ask, okay, well, which markers like to live next to each other, and which markers, which cell types like to live next to each other. And so this is a spatial proximity analysis that Liat pioneered, and basically how it works is you basically take a tissue that you're looking at, you'll hold one marker's location fixed, and then you'll take another marker and randomly permute like which cell types, which cells that marker lives in. And then you can then ask, okay, during this random permutation, how often do these two markers live in close spatial proximity? And you can build up a histogram. And so this basically lets you ask, okay, well, if I have two different markers that are living next to each other, is this a real interaction, or is this something that could just happen by chance? And so you basically use this approach. You can get a z-score, which basically tells you like how is this like something real, or is this something that just likely to occur by chance. And so you can do this for every pair of markers that we looked at. And so what we saw is that they're basically, when you compute z-scores between every pair of markers and then you do some clustering, they're basically two different patterns that you see. And so this is patient number 16 on the left, and then patient number 2 on the right. And so what you see is that on patient number 16, there are basically like two very well-defined clusters here highlighted in blue and highlighted in orange, whereas on patient number 2, there's really no significant clustering at all. And so what this represents is clustering of immune cells and tumor cells into their own compartment. And so you can then just go and look at, you can then go and look at what the images look like. And so these are these images with the immune cells colored in orange and the tumor cells colored in blue. And so we see what these represent is that on patient number 16, it's really that the tumor cells start forming their own compartment and the immune cells start forming their own compartment. Whereas the patient number 2 on the right, there's really intermixing between the tumor cells and the immune cells. And for completeness, there are actually 3 different patterns. One called cold, where there isn't really much immune cell presence at all. This is the middle one, which we're called is infiltrating, where there's just very fine mixing between the immune cells and the tumor cells, and then compartmentalization, where the tumor cells and immune cells form their own compartment. And the very interesting thing is that this compartmentalization or this broader tissue architecture is correlated with immune checkpoint marker expression. And so this is what is shown on this plots. And so the takeaway is that for here, PD-1, in patients that have this mixed phenotype, PD-1 likes to live on the CD8 positive T cells. And for patients that have compartmentalized phenotype, PD-1 likes to live in the CD4 positive T cells. And there's a similar story with PD-L1 living on tumor cells and immune cells. And lastly, this cohort of patient actually did have some survival data. And what Liat was able to do is ask, like, okay, well, of the patients that fall into different groups, how well do they do? And what she found is that the patients who had compartmentalized tissue architectures, they actually lived significantly longer, the patients that had the infiltrating or mixed phenotype. Unfortunately, there's very little data on patients who had a cold phenotype. And so that's actually where we're at. And so our takeaway is that the tumor microenvironment is complicated, but we now have the technological tools, both on the measurement side and also the computer vision side, to actually start disentangling that complexity. And so from our study, like what we're able to see is that the immune checkpoint markers are co-expressed both at the single cell and the single patient level. These checkpoint marker expression is correlated with tissue architecture. Tissue architecture is correlated with survival. And so what we're working on now, expanding this approach to new cohorts, we're actively working on improving our deployment of these machine learning computer vision models, and also jointly working to develop models that have new abilities. So we showed nuclear segmentation. It'd be great to do other cellular cell compartments, and we're actively working to sort of automate our ability to get these data sets curated, and also be able to handle both 3D and 4D data sets. And so that's it. Definitely want to thank everybody for tuning in. Definitely want to thank Michael. It's been a great collaboration. I have to thank all the members of my own lab who have been working night and day to get this stuff done. All the members of the Angela lab, who I not have time to mention, and also a couple other labs that we collaborate with, the Covert lab and the Chi lab for their contribution as well. And lastly, I need to thank our funders. So this work was funded by the NIH, the Paul Allen Family Foundation, Burroughs Wellcome Fund, an AI for Everybody Award from Figure 8, and also the Division of Biology and Bioengineering at Caltech. And so that's it, and I will open up for questions.
All right, thank you so much. That was fantastic. We've had a few questions come in, and we're running a little bit late on time, so I'm just going to stick with a couple here. One is fairly straightforward. Someone asks, is the software online from that URL, and this was a couple slides back, is it open and free to use for any purpose? And this person is just asking about the software you're using to implement deep convolutional neural networks. Can you talk about that?
Yes. So we basically put together a library of tools that we term DeepCell for doing like all of this work. And we're actively working on making that library sort of better and have like the newest, latest, and greatest, both as far as the actual like neural network architectures, as far as the different pieces of code that you need to pipe data into like your neural network training pipeline, and as far as deployment, we're also putting together like a user interface, auto-generated Jupyter notebooks, et cetera, et cetera. So that newest version is like we're actively working on it, and so it's right now like that's closed. We do have a GitHub repo. I believe it's github.com/vanballen/deepcell-tf, which basically has like implementations of like code that was used for this purposes. It is open source as far as completely free to use. I would say that the licensing right now is yes, but that might change. What we're likely looking at is a model where it's free for academic users to use, but commercial entities would likely need to contribute something to help further like the development. But yeah, open source and just letting everybody see like the code base, sort of like the nuts and bolts of like what we're doing, absolutely. And as far as having things viewable, I'd say like stay tuned. We'll probably have something released like within the next month.
Okay, great. A similar question, someone asks about data sets, and do you know of any good sites for getting X-rays or MRI data sets for deep learning?
So X-rays and MRIs and CT scans, I do not know the exact websites off the top of my head, but I know for X-rays and CT scans, the NIH has put together curated data sets for those. And I don't know off the top of my head where to get access to them. For CT scans, I know one of the prior Kaggle competitions was lung nodule identification. And so I'm pretty sure like Kaggle's website probably has the old data sets for CT scans, chest CTs. I believe the NIH recently released a newer, bigger data set, but I would basically, do a Google search, chest X-ray NIH data set and CT scan NIH data set. MRIs, for sure, they're out there, but I am less familiar with where those would be.
Okay, great. We'll go with one last one. So you touched on this a little bit, but someone asks, in deep learning, one of the most important factors determining results of our model is feature extraction from image data. What are the feature extraction techniques used in medical images, like MRI images?
So I would say they really fall down into two different schemas. So one is feature engineering and the other is like representation learning. And so feature engineering is you and a few of your friends get together and just brainstorm, okay, what are the features that we think we need to pay attention to? And so for this cell segmentation task, it could be like, hey, we should be looking for edges because that's the difference between anterior and background. You could be looking at particular textures or some collection of those. And those are really things that you have to like sit down and just think about what they should be. The other approach is representation learning, and that's really, I'd say like that's the space that I consider like deep learning to be in, is learning like what features should you be paying attention to be able to do your task well. Yeah. Which feature? Yeah, and so it depends on like when you can use one versus the other. If you have a lot of labeled data, then you can use the deep learning approaches. But as you saw from this talk, even then, there's likely like feature engineering that you're doing like on top of that. And so for our work, the features that we cared about was, okay, like we use the deep learning and the representation learning to get an idea of like where all the cells are, but then there's additional work that has to happen after that. And so what we're interested in was, okay, well, what are the different cell types that are there? What are the different cell states that are there? Who's living next to who? Like these were the features that we cared about, and we had to do feature engineering to extract that information. And so, for instance, identifying the cell types, we basically use the segmentation masks to pull out the different pull out expression levels of all the different markers. And then that was our quote unquote feature. And then we essentially use self-organizing maps and hierarchical clustering to sort of group things into clusters that made sense. Things weren't perfect, but that was sort of, like the feature engineering that we had to do in this work. But it varies very, very much from, sort of like what your imaging types looks at and what sorts of questions you're trying to answer.